that the citric acid cycle is both catabolic and anabolic, and we had just finished up catabolism, which is the breakdown, so from larger things to smaller things. In particular, we've discussed how we could go from carbohydrates ultimately to pyruvate. Pyruvate can then enter in. Of course, it's like Moses with the Holy Land. Pyruvate can see the citric acid cycle but can't enter in directly. It has to be transformed into acetyl-CoA, which was the answer to one of the questions. Because it's acetyl-CoA, it's a product of the pyruvate dehydrogenase complex. And you can also break down proteins. The easiest one to see is alanine, because alanine is the amino acid form of pyruvate. Pyruvate is the keto acid form of alanine, whichever way, tomato, tomato. And likewise, you could also see it with, oops, wrong one, sorry. Likewise, you could also see it with, where is it? Glutamate going to alpha-ketoglutarate, or, where did we go? Aspartate going to oxaloacetate. So, and of course, fats. What you'll find out in just a chapter or two down the road is that fats get broken down, the fatty acids get broken down directly to form acetyl-CoA, and so it's how they can enter directly into the citric acid cycle. Okay, so now we're going to start talking about how citric acid cycle is used for anabolism. Well, this actually goes hand in hand with another question that on the quiz today, which I kind of like, because I'm looking for the logic. And that's knowing what you know so far, which diet is more detrimental, one that lacks glutamate or one that lacks isoleucine. And then explain, what do you think, which one is going to be more difficult for you to recompense, to get over, as if you don't eat glutamate or if you don't eat isoleucine? Isoleucine. Ooh, we got like a debate going on. Fight. And so, why glutamate? This is one of the reasons why I had you explain your rationale. But sometimes you may get the answer wrong but have the right rationale. Why do you think glutamate would be more detrimental?
Charged, reactive.
No. So what rationale did someone put down for glutamate?
I completely guessed, but I said something to the extent of like that isoleucine, and then like you have leucine too, so like you could have different like isoforms of it.
Okay. Did anybody else? What did somebody else say who answered glutamate?
I said that glutamate was a keto form of alpha-ketoglutarate, which is used in the citric acid cycle to create energy, so without it, it could lead to low energy levels.
Okay. See, now that's good rationale. What?
Instead, I like that answer better.
Did someone say isoleucine?
I didn't say it on the quiz, but I'm pretty sure that's the one it is.
Did anybody say isoleucine?
I did.
What?
For a good reason.
Oh.
Because we need it.
It is. It is. We do need it. It is essential. We can't synthesize it. Because if you remember back from last semester, it's a branched chain amino acid. Plus, if anyone who works out a lot, you know you have to get your BCAAs. It is a branched chain amino acid, and it's more difficult to synthesize. One of the reasons why it's also the answer is actually for the reason that Hannah just gave, is the fact that we can actually synthesize glutamate from alpha-ketoglutarate, and you can get alpha-ketoglutarate from fats and from other things, since you can feed into the citric acid cycle, and the citric acid cycle can be used in anabolism to make glutamate. And so that's one of the reasons why I could have asked the same thing, like, you know, alanine, aspartate, glutamate, those are ones that directly come from the citric acid cycle or can feed into the citric acid cycle. So that's why it's this whole idea that there's this homeostasis to where if you start getting a little low on glutamate, you can scavenge, to an extent, obviously, to an extent. You still have to have other dietary restrictions, but you can scavenge it from alpha-ketoglutarate, and alpha-ketoglutarate can come via the citric acid cycle. And you can get the stuff from the citric acid cycle from other carbohydrates and from fats. Okay?
Wait, so the answer is glutamate?
The answer is isoleucine.
Oh. Okay.
You had the right answer. She had the right rationale.
I thought you were asking if I put that down. I was like,
No.
Right. So isoleucine is actually more detrimental because we have a way to make glutamate if need be.
Ask us those questions. Any of the ones we can synthesize are not the right answers because we can get them from other stuff.
Any of the ones that we can synthesize
From like alpha-ketoglutarate.
to make glutamate.
Yeah, and then
Right, or like if alanine was a choice, yeah, you can actually have a low alanine diet and still live and be fine because you just eat a bunch of sugar, the sugar goes down to pyruvate then can become alanine.
So basically, branch chain, we have to eat other
It's
ones.
right. Well, I mean, it's not just a branch chain. I think methionine is one that you really need. That's one I can think of off the top of my head. Phenylalanine, and so there are some of the others that are essential. I mean, the essential amino acids because we don't have a direct way to make them. Okay. All right, but this is actually really a review. So I've already talked about this particular slide multiple times. And so the citric acid cycle can be the starting source for other compounds, including both glutamate and aspartate. And so what kind of enzyme or enzymes can do this? What class?
Aminotransfer.
It's aminotransferases. Okay, and we're going to find out when we talk about the amino acids and the biosynthesis and the oxidation of amino acids is glutamate plays a major role, and alpha-ketoglutarate plays a major role in all of the aminotransferases. But we're going to see that a lot in the future. Yeah, that transamination, which was so great, but it's aminotransferases. Okay, and likewise, if you take something out, so let's say that we used up an alpha-ketoglutarate to make the glutamate, you're going to have to replace it. And so you can replace it by pyruvate, which pyruvate came from glucose or from even fructose or one of the feeder pathways into glycolysis. So that's why I said that you would have to, in order to stay alive, you'd have to have eaten your sugars or your fats or something in order to ultimately make the glutamate. But that's how they are all interlinked. Okay, and so this is just showing oxaloacetate. Of course, we've seen pyruvate carboxylase. This is actually part of gluconeogenesis. Remember, this was the first step of gluconeogenesis. But it could then be used to make oxaloacetate. Oxaloacetate can then go around the circle to make the alpha-ketoglutarate if you were needing to replace the alpha-ketoglutarate or any of the in-between metabolites. Succinate, fumarate, malate. Okay, this came out of the old book, but it does show like how what mammals can do versus some of the other organisms. In particular, how we can go from pyruvate directly to oxaloacetate, because that's the carboxylase, pyruvate carboxylase. However, there are some organisms that can actually go directly from acetyl-CoA to make oxaloacetate. We just can't do that. Okay, but there are just ways to supply the metabolic intermediates as needed or if needed. Okay, and so this is our little happy mitochondria once again. So this just shows how we've got this citric acid cycle going, and how once you get the citric acid cycle going, if you need to make carbohydrates, so gluconeogenesis is you can have malate go through the malate shuttle, the reversal of malate dehydrogenase to make OAA, and then that's just gluconeogenesis to make your carbohydrates. Or if we needed to build protein, this isn't the, I knew it. If you needed to build protein, or you get, say, glutamate for the glutaminergic neuron transmission, then you could even go this direction for the citric acid cycle to alpha-ketoglutarate, and then use aminotransferase to make your glutamate to go on and do whatever you need to do with glutamate, just as an example. You can ignore where it says typo, because I fixed the typo. And so you can also use build up lipids. And so we're going to find that out in, I forgot what chapter, for lipid biosynthesis. But lipid biosynthesis takes place in the cytosol, whereas once again, the citric acid cycle takes place in the mitochondrial matrix. Okay, and so you have to do it indirectly. So that what they do indirectly is you synthesize the oxaloacetate from citrate, okay? And what this will do is it actually will make NADPH instead of the NADH. It's just specific for NADPH rather than NADH. So this is one way that we can also make NADPH, but what's the main way? Does anyone remember from earlier? What's our main source of making NADPH? The pentose phosphate pathway. That's the primary source for it, but this is another backup. And out of that you get acetyl-CoA, and acetyl-CoA is the building block to make the fatty acids, since you add them on 2 at a time for the even-numbered fatty acids, which most of them are even-numbered. Okay. Essentially, that's what I was saying here, is inside the mitochondria, you were utilizing NADH with the malate dehydrogenase, but the cytosolic form of it, you're ultimately going to get NADPH out of it. Okay. And so even though the entire process starts in the mitochondrial matrix, you can use these shuttles to effectively transport electrons and transport other biomolecules from the matrix to the cytosol without, or even if they, electrons wouldn't just freely go through. And so what you do is NADH and NADPH can't, and so that's why you use these dehydrogenase enzymes that effectively carry the electrons from one side to the other side, and then ultimately would, regenerate the NADH or the NADPH as needed. There it is. Okay, so this is the big summary. Boy, that did not come out very well. It looks much better on here. It looks very jaundice there. But I really like this because it kind of shows how everything is tied together. Some of the way that we did catabolism, not just carbohydrates vis-a-vis the gluconeogenesis, but we can also then go on and use it to help get some of the amino acids. We can use it to get some of the new nucleotides, and including the NADPH necessary for the nucleotide development. And then, which we will not learn how to synthesize every single amino acid that's possible. Okay, we'll learn a handful of the important ones or how they are used, but it's just, you could have course after course on just amino acid biosynthesis and their metabolic pathways. And likewise, we can use to make our fatty acids and steroids. Okay. Now, not every organism has to undergo all of the citric acid cycle. So especially the seeds on plants, and there are some other microbes that can use what's called some books call it the glyoxylate shunt, some call it the cycle. And essentially, it effectively will skip over decarboxylation. And I'll show you, I've got a better picture that shows that in just a moment. Especially a better picture than that right there. And so it bypasses some of the steps of the citric acid cycle. Right? And so the plants use a special compartment called the glyoxysome where this can occur, which is really important, like I said, for seeds. So you can go through glycolysis or the breakdown of fats to get your acetyl-CoA, and it starts through the citric acid cycle, but once it gets to isocitrate, it then uses a lyase to cut it all up into 2 carbon-containing glyoxylates. And these glyoxylates then hook up with another acetyl-CoA in order to make malate. So effectively, you completely skip steps 3 through 5, but you still get your NADH, which is needed for to make ATP. So I've got a better picture that It may not be as pretty, but I think it's easier to read. Oh, well,
Is there some state
all I know is it's like haunted. I'm getting like a subliminal
Biochem
message.
rave.
I don't know about you guys, but I'm picking up a subliminal message from the projector.
No, I think it's a biochem rave.
Images are showing, just can't see it.
And so, I should have hidden these and had you guys guess, but yeah, this is isocitrate lyase. So it goes from having 6 carbons to 3 2-carbon containing units. And then this is a synthase, which what's the difference between a synthase versus a synthetase?
Taste uses energy.
Right, synthetase uses the external energy from like ATP, GTP, things like that, whereas the synthase does not. And this is malate synthase, which takes, because malate, if you remember, it has 4 carbons. It takes 2 carbons from acetyl-CoA, 2 carbons from the glyoxylate from the breakdown of isocitrate to make malate. And then it just goes ahead, and this is malate dehydrogenase, so we've seen like normal. And so by bypassing this, you don't have to worry about decarboxylation. Okay, so it's kind of like a way for like an energy resting stage in that sense. You don't have as many steps to go through. Yet you still get FAD, and you also pick up your FADH too, because you can get succinate out of this. Then I always try to bring this home to something biomedical, since most, if not all of you, are geared that way. And here, this is a picture of someone that's suffering from beriberi. And this is the kind of thing that I also like to put sometimes on medical implications on exams, where you have to try to explain like some of the phenotypes or why it makes sense, like some of the symptoms that some of these things have. Okay, so there are a couple of ways to develop beriberi, but one is just from diet. Okay, and where was this really noticed, really, I don't know, first isolated from? Does anyone know with beriberi and some of the related diseases?
Yeah.
Close. I mean, primarily in parts of Asia, and especially in Polynesia, in parts of Asia, where what happened was after the West had, I don't want to say invaded, but had introduced their diets. In particular, what's one staple of a lot of Polynesian and Asian diets?
Rice.
Rice. And they introduced this idea, instead of using wild rice or brown rice, to remove the husks, to where they developed and they were eating much more just white rice. Well, that lacked vitamin B1. And so then their diet was void of vitamin B1, since that's in the husk part of the rice kernel, or rice grain, I should say. And why does it make sense then that they would have some of these phenotypes where heart problems, malaise, so lack of energy, nerve damage, and you can eventually die from it? Why would that make sense?
Don't have enough energy.
They don't have enough energy because... Right, their citric acid cycle is just decreased. And that's because thiamine, which is one of the cofactors of the pyruvate dehydrogenase complex, which was also on the quiz today, thiamine is derived from vitamin B1. Okay, and so because of that, then they don't have an adequate amount of PDH or alpha-ketoglutarate dehydrogenase. So therefore, their citric acid cycle, their Krebs cycle, is deficient. So there are still ways to make NADH, but it's just not to the level that's necessary for life in the long term. Okay. All right. So are there any questions over that, and trying to start to use the logic on how, if you alter the homeostasis, the ratio of almost anything from one form to the other, how it's going to have to compensate that between these various metabolic pathways, and how these metabolic pathways interact with respect to each other. So we have gluconeogenesis coming from citric acid cycle, glycolysis going towards it, pentose phosphate pathway comes off of those two, things like that. We haven't covered amino acid oxidation or fatty acids yet, but we will. But are there any questions or comments, concerns? Monday is the exam, don't forget. And like I said, I'm going to try to post the quiz 4, which would just be to review online, and it won't be due until the exam time, so that way you can have plenty of time to work on it. But